like to invite Irina for her presentation. She's a senior researcher in the lab of soil carbon and microbial ecology, and the title of her presentation is Approaches for the Monitoring of the Soil Phytopathogens.
Okay, so it will be a bit common report. It will not be more data, it will not be data, but more theory, because I want to tell about what is pathogens and how we can work with it in the soil. So, as we know that plant and microbes is a big system which is very important for the living of the plant and also for the processes which goes in the soil, and we can distinguish the system in a different, according to the different parts of the plant. Like the top, it will be philosphere, stem and leaves. Endosphere will be inner plant of the plants, but now we are not interested in it, and it's very difficult system. Also, under the soil it will be rhizosphere and rhizoplan, which is more interesting for us. Rhizosphere, it is the soil which is around of the roots and includes connections between roots and exudates to the soil, and of course, and microbes. And rhizoplan, it is the direct surface of the roots. The plant, all this microbiome includes different types of organisms, which can be bacteria, fungi, oomycetes, like viruses, protists, algae, nematodes, also, of course, probably other soil animals, and so on. And they should live in a normal complex, which is good for and first and the second part of these interactions. But of course, as we know, that there can be pathogens which can change it all. First, in the past, it was that these interactions between pathogens and plants, it was like one pathogen, one plant, host plant. But in further, because of the knowledges, it was made the term path biome, which is this concept includes the totality of microbes which interact with with pathogen species, and how they influence to the pathogenesis at all. And for understand this path biome, we should make these goals, like accurately description of the biodiversity of these microbial communities. We should shift from description to functionality to understand how it works, how it influences, how path biome modulates pathogenesis, and how it impacts to the environment. We should understand the impact of microbial communities to the pathogenesis, also how to interrupt this, how to make a plant to survive this. And of course, we should improve knowledge about biotic and abiotic factors, which can be the factor who can start the process of pathogenesis. The pathogens can be distinguished to different. The pathogens are very different from different parts of the microbiome, and mostly it is fungi, which can create different types of the rods, like stem rods, the crown rod, also, of course, root rods, they can contaminate seeds and small young plants. And further, I will talk about it more because I prepared in the further slides. We can distinguish also fungal-like organisms like Mycota, and they also very important for distinguish pathogens. The smaller group is bacteria. It's not much, but of course they exist, like Erwinia or Streptomyces or something else. Also, it will be protozoa like nematodes, viruses. It's also not big group of pathogens, but of course they also, and as I remember it, there are enough also research about this. And I also included parasitic plants and insects because of course they are, but it's not our field of interest. So we continue. And I prepared a pack of the most important of pathogens which we can find on our fields, but of course it's not all. It's a huge amount of it. And I will start from Fusarium oxysporum, which is one of the most important. It's a soil-borne pathogen which causes different wields to the wide range of plants, and according to the knowledges that there is some strain of oxysporum which can be to the some species of the plant, and for this scientists made the different formae specialis like Betaea, Cucumberum, fragrae, and so on, and also tuberose, which is named according to the plant which attacks this Fusarium oxysporum. The next will be Fusarium graminearum, also Fusarium, but a bit more concrete. It mostly attacks crops like barley or other, and it is a huge problem for the different countries. And you can see that on the photo it creates that Fusarium head blight on the stems, and also you can see on photo C and D how different will be grain. Also, I prepared how it looks on the Petri dish, and also its macroconidia. Next will be Verticillium dalai and Verticillium alboaldrum. Sometimes it can be synonyms, but in some works it is different species, but they too pathogens cause very the verticillium wilt of many economically important crops, and you can see also how it attacks here the stems, how it attacks also the high plants. Also on the Petri dish you can see also the conidia, microconidia, also sclerotia, which it creates. Next, it's Botrytis cinerea, also very important pathogen. It creates grey mold on a lot of species of plants, but mostly it attacks strawberry and grape. Usually you can see this how it consequences of it on this photo, and you see that how it also creates sclerotia, which we can find. And it's very, very also widespread. Calitotrichum, it's one of the most important of genera of pathogenic fungi, and most of crop species can be a host of some of species of Calitotrichum. This fungi also you can see how it attacks different pepper or banana or other plants around the world. This species is Peruclaria arizaea, which is now it's renamed from Magnaporia arizaea, and it's a fungus which attacks rice, and it is a huge problem because also, as we know, that the huge part of the planet eat rice. So the important part to understand how we can protect plants from this pathogen. And you can see consequences when it attacks and how it influences to rice. Phytophthora is omicet pathogen, which this genus is also creates devastating diseases in the different crops, also very worldwide and very important. And many species can be and which can attack also woody plants and also crops. So it's really everywhere. Also the species Rhizoctonia solania, which is very important also for us because our work is based on a potato, so we can see that how it takes also tubers and the stems and leaves of potato and what it creates inside. It's also now there are a lot of studies about it, of course, because it's one of the worst pathogens for the worldwide agricultural sector. I will talk about how we can study these pathogens, and I will start from the previous mostly important method. It's morphological and genetic identification, because of course when we extract some strains from our samples or plants or soil, we should understand what it is. So we start from isolation. Of course, after we incubate, create few couches, we identify by morphology with microscope and some previous data, how it can be sclerotia, microconidia, or other parts of the fungus. And after, if we have it, we can preserve it in some collections or in our laboratory. But of course, this method is not enough, absolutely. And the problems is it depends mostly from the eyes of the scientists who study it and from the eyes of previous people who may create the keys of the fungal. So now we can afford to extract DNA and made PCR and sequence and all these processes and identify by DNA, by different parts of the DNA. For any species, it should be different. But of course, also this cannot be all, because the most important problem of this method at all, the two of these parts, is that we cannot cultivate that fungus which cannot be grown on our media. For this, we should use another. And this problem that we lose a lot of possibility to describe our pathogenic community, and not only pathogenic, but also, of course, all microbiological communities. So we can use the method real-time PCR, which is also polymerase chain reaction, but of course with the difference that we can also calculate directly how many DNA parts is in our samples. And the method is very useful when samples are small, and we also can make a lot of samples simultaneously, like 200-300, and we quantify how many different pathogens can be in our sample. But the most problem is that we, according to that, we should make a primer system which can start this process, and we can understand what we want to find. And of course, it can be some problems like interruption, some multi-reading. We can find some extra course here which cannot be our what we search. So of course we should use the complex of these methods. And another method is metagenomics, also should be included to the whole process of research. And metagenomics, of course, can help us to understand absolutely all what we can find in our samples. And we can start it for bacteria, fungi, and other organisms, but the most problems that we cannot say that what we find, is it what is really living here, or it is just some derived genes from other parts of the world. And we cannot we cannot say that it is really part of this direct sample. But of course, because of this method, we can understand what is taxonomic profiling, of what is what we can find here, and we can also understand functional groups of these organisms. But of course, we cannot use it without any different other methods. And when someone say that we can use just this method, and we cannot, and we don't need, for example, morphological identification, it's not good, because of course we should also see by our eyes and what we can grow on the petri dish. Thank you for that.
Thank you. Maybe one small question.
Thank you. Can you recommend some approach for express monitoring of endosphere pathogens? Example, club roots of cabbage. Is it genetic analysis?
I suppose yes, but of course we should understand that it should be very clean conditions to extract something from there, because we also cannot forget about derived genes. And I suppose if we cut some stem or some leaf and go to inside, it should be some very, very clear box. And after, yes, I suppose we can make DNA, and also we can also try to grow something on petri dish. It's not a problem. We just put the part of the cabbage on the media, like PDA or something. PDA probably is the best, because it's potato agar. So I suppose a lot of can grow on it. And to look what we, and after, yes, of course, because we can understand about, I think that the most important to understand which primer pairs we should use, because of course it's the plant, which the most of DNA will be the plant. So we should understand how to separate the plant and the microorganisms from there. Of course, for bacteria it's very simple, but for eukaryotes it will be much more difficult. And as we know also for fungi, that not only ITS like before we should sequencing, but also other like beta tubulin, like in other parts of genes, and it will be more complex research of it.
So it is a big problem.
I know. I suppose it's not a big problem. I suppose just we should be very careful what we search there inside.
Okay, thank you.
Mm.
Thank you.
So if no one has more questions, so thank you again for your presentation.